The overall goal of the following experiment is to identify culture conditions that best enrich for ovarian cancer tumor-initiating cells. This is achieved by first transferring the cells of interest to low-attachment flasks in serum-free stem cell media containing growth factors. As a second step, flow cytometry is performed to assess the CD133 expression and ALDH activity of the cells. Next, cells from the cultures that are most highly enriched for tumor-initiating cell markers are subcutaneously injected into immunocompromised mice to verify the tumorigenicity of the cultures. Ultimately, flow cytometry and subcutaneous xenograft models can be used to determine the culture conditions that elicit the highest percentage of ovarian cancer tumor-initiating cells.
We first had the idea for this method when we noticed that some of our cell lines had unusual growth characteristics and appeared to thrive as free-floating clusters, a phenotype that would likely resemble what occurs in vivo in ovarian cancer patients.
The implications of this technique extend towards the therapy of ovarian cancer, as there is a high rate of recurrent chemo-resistant disease in ovarian cancer, and much evidence suggests that tumor-initiating cells contribute to this phenomenon.
To generate a floater tumor-initiating cell culture, first transfer the floating cells of an 80% confluent cell culture into a 50 milliliter polypropylene centrifuge tube.